Today I am going to discuss about the topic the fertilization of angiospermous plant. So basically the fertilization process in angiospermous plants is the fusion between the male gamete and the female gamete. As we all know that the pollen grain which is produced within the pollen sac of the androecium, and then we also know that the egg cell which is produced within the ovary of the gynoecium, which is basically developed within the ovary of the gynoecium, the egg cells will be produced. The fusion between the male gamete and the egg cell is known as fertilization. In angiospermous plant, the actual fertilization process is going to occur within the embryo sac. The structure of an embryo sac is of an angiospermous plant is like this. The embryo sac, it is in 7-celled and 8-nucleate stage structure. It is 7-celled and 8-nucleate stage structure. Let us draw the proper structure of an endosperm. Here is 3 cells. There are also 3 cells. Okay, this portion or this pole is known as microbial end, and these poles are known as chalazal end. In this chalazal end, there are 3 cells. Both have nucleus, and in this microbial end, there are also 3 cells. They both have nucleus. The 3 cells in this chalazal end is known as antipodal cell. These are known as antipodal cells. And towards the microbial end, this cell is known as egg cell, and the another 2 are known as synergids. And in this central portion, there is a cell which is known as central cell. It is made up of 2 polar nuclei. Due to the fusion between 2 polar nuclei, this central cell is produced. The entire process of fertilization is going to take place within this embryo sac. This is the structure of an embryo sac, and the entire process of fertilization is going to occur within this embryo sac. Okay. Let us understand how does the process of fertilization is actually occurring within the angiospermous plant. For this, we have to draw the structure of the gynoecium of the angiospermous plant. This is the structure of a gynoecium. This portion is known as stigma. This long portion is known as style, and this portion is known as ovary. Within this ovary, there is a ovule, and within this ovule, the embryo sac which is present. In the process of pollination, the pollen grain basically landed in this stigma portion. Pollen grain is going to landed in this stigma portion. So the pollen grain is landed in this stigma portion. At the point of landing, the pollen grain is in 2-cell stage. Let us draw the structure of a pollen grain when it is landed in this stigma. This is 2-cell stage. The larger one is known as vegetative cell, and the smaller one is known as generative cell. Okay. Actually, the vegetative cell, it is going to develop the pollen tube. And the generative cell, after landing in this stigma portion, when the vegetative cell is going to develop the pollen tube, then this generative cell undergoes a division and form two male gamete. Okay. In the next step, we will observe that. Let us clear this portion and we will continue the process of fertilization. Okay. As the pollen tube is developed from this, pollen grain, it started moving forward. The pollen tube is going to start moving forward. Their basic aim is to enter into the embryo sac and to conduct the process of fertilization. This pollen tube is moving forward through this tile. They are moving forward. Moving forward. This is pollen tube and this is moving forward to enter into the embryo sac. Let us draw the proper structure of an embryo sac again. This is the structure of embryo sac. Here are three cells. The cells are known as antipodal cell through the chalaza length. To the chalaza length, these three cells are known as antipodal cell. Here towards the micropylar end, there are also three cells. One is known as X cell and another two are known as synergids. And in the central portion, there is a cell that is known as central cell. It's known as central cell. As the pollen tube reaches the embryo sac, it will enter into this embryo sac. This pollen tube is already containing two male gametes. It is already containing two male gametes. The synergid cell. This is the synergid. Synergid will give instruction to this pollen tube to enter into the embryo sac. Through the synergid, the pollen tube enters into the embryo sac. After entering into this, the two male gamete will get free. This is one male gamete, this is one male gamete. Now these two male gametes are going to fertilize. One is going to fertilize the egg cell and the another one is going to fertilize the central cell. One is going to fertilize the egg cell and another one is going to fertilize the central cell. As we can observe here, there is fertilization occurring in two times. Fertilization is occurring two times. First time, the one male gamete is going to fertilize the egg cell. Second time, the male gamete is going to fertilize the central cell. So, in this angiospermous plant, the fertilization is also known as double fertilization. This is known as double fertilization. First process of fertilization, one male gamete is fertilizing with the egg cell, and then the structure which will be produced is known as zygote. The male gamete is in haploid nature, the egg cell is also in haploid nature. The zygote which will be produced will be diploid in nature. The second fertilization is occurring between one male gamete plus central cell. This male gamete is haploid in nature, but the central cell is 2n in nature, that is diploid in nature due to the fusion of two polar nuclei. This will develop the structure known as endosperm. Endosperm. This endosperm is triploid in nature. N plus 2N, that is triploid in nature. The fertilization is occurring in two phases or two times, so it is known as double fertilization. And due to the second fertilization, as the endosperm is produced, which is a triploid cell, so this process is also known as triple fusion. Double fertilization and triple fusion. In angiospermous plant, the process of fertilization is known as double fertilization and triple fusion.